Good morning, everyone. Is it visible?
হ্যাঁ স্যার দেখা যাচ্ছে, কিন্তু স্যার আপনার কথাটা না একটু আস্তে শোনা যাচ্ছে।
Hello, hello.
হ্যাঁ হ্যাঁ এবার শোনা যাচ্ছে ঠিক।
Okay, okay, okay. Thank you. So last day I have started started the bioelectrodes. So today I would like to discuss about the bioelectrodes, working principles, and what are the different types of electrodes are available. So let's start. So before going to discuss in details the bioelectrodes, first we have to know the electrode electrolyte interface. So last day I have discussed what is the function of bioelectrodes. Basically, uh, um, electrodes these are uh, used as a transducer. Okay, and the function is the current as it is carried out the currents in the body through ions. So basically these electrodes are used to change, it's a transducer that is used to change an ionic current into an electric current. Okay, so electrode here, you can see in this diagram, this is the part of electrode and this is the electrolyte. That means in our body, which is combinations of different electrochemical fluids. So it's a electrolyte solution. So first, how this can be interfaced. So electrode and electrolyte. So electrons basically moving in a direction opposite to that current in the electrode. So here, so I current flows in this direction, and the electrons moving in the opposite direction. Okay, here. Now cations, these are moving, actually electrode, that is the combinations of metal atom, C, as you can see in this picture. So cations, these are moving in the electrolyte solution, these are moving in the same direction as in the uh, current. Okay, and but the anions, that is moving in opposite direction of the current, because electrode, that is the combinations of the metal atoms, but electrolyte solution, that is the combinations of different cations and anions. So these are, so there are actually no free electrons for charge to cross this interface. That is, no free electrons in electrolyte, or no free cations or anions in the electrode. Therefore the chemical reaction has been established in this interface. And here these are the chemical reactions. So basically electrode, that is made up of some atoms of the same material as the cations, and this material in the electrode at the interface, that can be oxidized to form a cation and some free electrons. And the cation, that is discharged into this electrolyte solution, and the electron, that serves as a charge carrier in this electrode. And when this anion coming to the electrode-electrolyte interface, that can be oxidized to a neutral atom after giving one or more free electron to the electrode. So here, as you can see in this diagram, these are all uh, reversible. These reactions are reversible, and these reactions occur even when no current is crossing the electrode electrolyte interface. However, the net transfer of charge across this interface, that is zero, as the rate of oxidation reactions equal to the rate of reduction reactions when current is crossing the interface. So uh, if the current, if this current flow, that is from electrode to electrolyte, the oxidation reaction dominate. But when the current is in the opposite direction, the reduction reactions dominate. Okay, so oxidation and reaction will be there. So here the different frequency range, those are applicable in case of ECG monitoring, or EEG, or EOG, or EMG monitoring cases. Now let's move on to the next slide, where there are some different properties of bioelectrodes has been enlisted. So the first property, which is that should be conductors, it should be low impedance, and it should not polarize when a current flows through them. And good contact, it must be good contact with the body and not cause any motion artifacts. And it should not causing itching or swelling or discomfort to the patient. It must not create some irritation to the patient. And metal, the electrode by which the metal by which the electrode has been designed and has been made up, that should not be toxic, that should not be reactive anymore. And it should be mechanically rugged, and must be uh, there must be some process where it is easy to clean. Okay, because every time some of the electrodes needs to clean every time after use, okay, for reusing purpose. So these are the different properties of the bioelectrodes. Now the electrode behavior of the circuit model. So here, this portion, the left side, that is the metal portion, and this is the electrolyte portion. This is the electrode, and so there is an interface. So if it is converted into an equivalent circuit, so this is the circuit. So basically bipotential electrodes that have the metal electrolyte interface, as I have told you earlier, and the electrode potential is developed across the interface, which is proportional to the uh, exchange of ions between the metal electrolytes of the body. Now this potential, actually it is called the half cell potential. I will discuss it after that. So the double layer of charge at the interface, it serves as the capacitor. Okay, and here, so this is the equivalent circuit of the potential bipotential electrode in contact with the body, that is comprised of voltage in series with the resistance capacitance network. Okay, and the DC voltage on account of this difference in electrode potential, that is known as the electrode offset voltage. Okay, and if the so sometimes, actually it is a single electrode electrolyte interface diagram, but there are certain most of the time we need two electrodes at the same time, two electrodes. Okay, so uh, because one electrode is not sufficient to measure or monitoring the actual bipotential to the measuring device, so that's why we need the two electrodes. And when the both electrodes being of the same material, the electrode offset voltage, that must be very small. Okay, and if both the electrodes have different materials, the electrode offset voltage is rather, it is also significant in this case. So the chemical activity that takes place within an electrode, that may cause the voltage fluctuations, and may appear as noise on a bioelectric signal. Okay, and this noise that can be reduced by proper choice of the material, or by special treatments, such as like coating the electrode by some electrolytic method. Okay, and such type of electrode, that is prepared by electrolytically coating a piece of pure silver with the silver chloride. Okay, so basically that is the electrode impedance, this resistance, and this RD and CD. It is the impedance of that electrode, and that is the series resistance of this electrolyte solution. And this is the half cell potential, that is the uh, voltage which is generated. So here, in this picture, right hand side, so it is a uh, first first we need to understand what are the layers are there in our under our skin. So basically the skin consists of three layers, which surround the body to protect it from the environment. So, and also it is an appropriate interface. The outermost layer, which is known as the epidermis. It plays a important role in the electrode skin interface, and layers consisting of three sublayers which constantly keep on reviewing that itself. It also situated here in this position, and the epidermis, that is the constantly changing layer of the skin. The outer surface consists of also the dead material, uh, having different characteristics from the live tissue. Okay, and the layers down below this epidermis, this layer contains the vascular and nervous components of the skin. In addition, it have some sweat glands and ducts and hair follicles. So the sweat glands do not follow any unique electrical characteristics of the skin. So under this epidermis, there is the dermis, a subcutaneous layer is also available. So in this diagram, actually, so this is our skin, and on that there is a electrolytic gel must be used for proper interface, and here is an electrode. So actually, there are two interface. Okay, one is the electrode and electrolytic gel, and another interface is between the electrolytic gel and the skin or tissue. Okay, so the equivalent circuit is given here. So the electrode and electrolytic gel interface equivalent circuit diagram is given here, this position. Okay, as I have shown you the last slide, like two resistance and this capacitor creates an impedance of this electrode. Here is the electrolytic resistance, and and after that, for the second interface level, it has another equivalent circuit, which is the combinations of this epidermis as well as uh, the uh, the dermis and the subcutaneous layer resistance also here. So why we need to electrolytic gel? Actually, this interface between the electrode and electrolyte and the skin, that should be um, understood to have clear picture of the behavior of the electrodes, and a transparent electrolytic gel which contains the chlorine as the principal anion that is used to couple an electrode to our skin, and that this gel also gives a good contact. Okay, it is used for conducting purpose. Similarly, and electrode uh, cream containing this chlorine, that may also be used for different biosignal monitoring purpose. So here in this uh, RS, actually this one, this is actually the effective resistance associated with the interface effects of this gel between the electrode and the skin. And RSE, the lower resistance, that is here, the it it creates some potential difference due to ionic concentration across the epidermis, or at least the stratum corneum as a semi-permeable membrane. Okay, so for 1 centimeter square skin, the impedance should be reduces from 200 kilo ohm at 1 hertz to 200 ohm at 1 megahertz. That is for 1 centimeter square skin. Okay, and the fluid which is secreted by these sweat glands contains this sodium, uh, potassium, chlorine ions, and concentrations differ from those in extracellular fluid. And there is a so there must be some potential difference between the lumen of the sweat duct and the dermis and the subcutaneous layers. And there are also a parallel resistance and capacitance combinations in series with this potential that represents the wall of the sweat glands and duct. So these components are generally neglected when we consider bipotential electrode. We have to consider only these two portions. Okay. So if a part of the electrode is in electrolyte and one moves, all of the other remains stationary, a potential difference appears between these two electrodes during this movement, and this potential that is called that is the bipotential, but it is actually one type of motion artifact which can be a serious cause of interference in the measurement of biopotential. And these motion artifacts actually results primarily from the mechanical disturbances of the distribution of charge at this electrode electrolyte interface, and it is reasonable to expect that the motion artifact is minimal for non-polarized electrodes. Okay, so move on to the next slide. Half cell potential, as I have told you, that the potential which is occurred between these two electrodes, that is called the half cell potential, and that is caused by the ability of electrons to flow from one half cell to the another. So electrons, as already discussed, so half cell potential, that is uh, determined by these three ways. Metal must be involved, and the concentration of its ion in solution must be considered, and the temperature of the surrounding environment. And a second electrode is required to find this half cell potential, which is basically a hydrogen absorbed in platinum black. So this hydrogen electrode is also needed. So in half cell potential, the Nernst equations, it is used to find out the different ionic activity. So when two ionic solutions of different concentrations are separated by a semi-permeable membrane, an electric potential exists across this membrane, which is equivalent to this minus RT by NF log rhythm of A1 by A2, where A1 and A2 are the ionic activity of the ions on each side of this membrane. And this ionic activity, that is the availability of an ionic species in solution to enter into a reaction, okay, which I have considered in first line. Now polarization, actually for uh, standard half cell potential, this polarization should be occur when current flows, the half cell potential is zero, that also be changes. Okay, and there was some over potential where this is the difference between the non-zero current and zero current half cell potential, and this uh, over potential, it is the combinations of this ohmic over potential, concentration over potential, and the activation over potential. Now what is ohmic over potential? It is happened due to the resistance of the electrolyte, which is the voltage drop along the path of this ionic flow. Concentration over potential, Vc, that is due to a redistribution of the ions in the vicinity of the electrode electrolyte interface. Basically this is the concentration change. And the activation over potential, which is Va, that is due to metal ions going into the solution, and or due to metal plating out of the solution onto the electrode. So there are two types of polarizable electrodes. One is the perfectly polarizable electrodes, and the second is the perfectly non-polarizable electrode. So perfectly polarizable electrodes are those in which no actual charge crosses the interface, electrode electrolyte interface, when current is applied. Okay, and that current across this interface, it is a displacement current, and the electrode behaves like a capacitor. Okay, as per example, platinum electrode. In case of perfectly non-polarizable electrode, so here the current passes freely across the electrode electrolyte interface, and it requires no energy to make this transition. So, and these electrodes, see, no over potentials. Okay, example, silver chloride electrode. Okay, and silver chloride electrode, that is basically used to a recording purpose. Okay, because it is easier to transfer the signal from that portion to the measuring device, whereas in this platinum electrode, that is used for stimulation purpose. Okay, now different motion artifacts can be found. So a blurring of a radiographic image produced by respiratory or muscular or other movement of the patient, the signal can be blurred. When polarizable electrode, that is in contact with an electrolyte, a double layer of charge forms the interface, which also creates a motion artifacts. And movement of the electrode, that will disturb the distribution of this charge and results in a momentary charge change in the half cell potential until equilibrium is reached again. Okay, and signal due to motion that has low frequency, so it can be filtered out when measuring a biological signal in high frequency components such as EMG, means to monitor the muscle signals. Okay, or action action potential, to measuring this action potential for action. For ECG, EEG, and EOG, whose frequencies are low, it is recommended to use non-polarizable electrode to avoid signals due to motion artifacts. Now coming to the next slide, which is different, that is for different electrode types, how many electrodes are available. So there are four types of electrodes available: surface electrode, micro electrode, internal electrode, and needle electrodes. So basically, internal and needle both are same type of, mostly same in nature. So under surface electrode, there are three electrodes: metal plate electrode. Three types of electrodes are available: metal plate, floating electrodes, and the flexible. So primarily it is, so for surface electrode, it is used in ECG, EEG, or uh, EMG purpose, because these are used to get the bioelectric potential from the surface of the body, and these are available in different sizes and forms. So basically, generally larger electrodes, these are generally used in ECG, as localization of the measurement is not so important. Smaller electrodes are suitable for EEG or EMG purpose. Okay, so it has different metal, suction, floating, and flexible electrode. So metal electrode, where that is used for application to limbs. Okay, it is used in our limbs. So metal disc electrode, that is used with surgical tape. Disposable foam pad electrodes are frequently used with the electrocardiographic monitoring equipment. Okay, and this disposable foam pad, as you can see in this picture, this is uh, this is a type of electrode which consists of a large disc of plastic foam material with silver plated disc on one side, which is attached to a silver plated snap similar to that used on clothing in the center of the other side. Okay, next is the suction type, where this is the metallic suction electrode. It is a modification of the metal plate electrode that requires no steps or adhesive for holding it in the place. So it consists of a hollow metallic cylindrical electrode which makes contact with the skin as its base. Basically, using this type of suction type electrodes, it creates irritation to the patient. Okay, because there is no electrolytic gel has been, or the adhesive is used between uh, the interface of the skin and the electrode. It is just suction of air. Okay, then next is the floating type electrode. So surface electrode, basically These are actually recessed electrodes with top hat structure like this in this picture here. Uh, Cross sectional view is given. The electrode that is also the same type of the feature, and another cross sectional view is available here. Okay, so it is formed from an open foam disc which is saturated with electrolytic gel and is placed over the metal electrode. Next is the flexible electrode. So these are these are carbon filled silicon rubber electrode and flexible thin film neonatal electrode. Okay, so cross sectional view is here. A pin connector is used basically is that is used to pushed into the lead connector hole. Okay, and these type of flexible electrodes, these are important for monitoring the premature infants. It is basically neonatal for treatment, for neonatal treatment. Then coming to the micro electrode, basically these have tips small enough to penetrate a single cell, and as such readings from within single cell that can be obtained. Okay, and these microelectrodes are basically two types of electrodes are available. One is the metal electrode and another is the micropipette electrode. So metal microelectrodes are constructed by the electro electronically etching the tip of a fine tungsten or stainless steel wire to the desired size. Subsequently, the wire is coated almost to the tip with insulating material. So here special electrolytic processing is also done on the tip to lower the impedance, and the iron metal interface happens at the point of contact between the metal tip and the electrolytes either inside or outside the cell. Okay, so here this is a diagram of a micro pipette type of electrode, which is actually a glass uh, micropipette with the tip of the desired size about 1 micron. This 1 micron. Okay, so the micropipette that is filled with an electrolyte suitable with cellular fluids, and such micropipettes is having dual interface. One interface is comprised of a metal wire in contact with the electrolyte solution inside the micropipette, and the other is the interface between the electrolyte inside the pipette and the fluid inside or immediately outside of this cell. Okay, and a thin film of precious metal that is bonded to the outside of a drawn glass microelectrode, and such microelectrodes are very reliable and having very good performance. Okay, and the impedance of this type of microelectrode is in mega ohms. Naturally, this demands amplifiers of very high impedance to avoid loading of the circuits. Yeah. Next is the internal electrodes. So here, for detecting the fetal uh, electrocardiogram during labor, so by means of intracutaneous needles. So here no electrolyte skin interface is there and no electrolytic gel is required. Okay, it's a one type of uh, needle and wire electrode for the intercutaneous measurement of this of biopotentials. And these are basically insulated needle electrode, coaxial needle electrode, or maybe bipolar coaxial electrode and fine wire electrode that is connected to the hypodermic needle before being inserted into our skin. Okay, so here cross sectional view is there, uh, and the view, so it is view of this electrode and the skin interface. So muscle with the coil fine wire electrode in, it is placed on the skin. The needle is usually made of stainless steel and is insulated with insulating varnish. Only the tip is left exposed, and these are basically used in electromyography. Okay. So next is the needle electrodes, where it penetrates the skin to record the biopotentials, and also it reduces the interface impedance. So single wire inside the needle which acts as the unipolar electrode that is used to measure the potential at the point of contact where it is applied. And there are different types of needle electrodes, like concentric, like bipolar, or the monopolar needle electrodes are available. So what is the application of these bioelectrodes? It can be used in cardiac monitoring purpose. It can be used for infant cardiopulmonary monitoring purpose, mm, sleep encephalography, or it can be used to diagnostic a different treatment related to the muscle activity, or for ECG purpose, or it can be implanted telemetry of biopotentials that can be transferred the signals from one position to another positions. uh, and it can be used for eye movement, to monitor this eye movement also. So I need to I also discuss what is the practicability in using these electrodes. So no parts of this lead wire that should be exposed to the electrolyte, all of the same material. A third material, that says solids, should not be used to connect this electrode to its lead wire unless it is certain that this material will not be in contact with the electrolyte. And this lead wire that can be welded to electrodes, or from a mechanical bond through crimping or the pinning technique. So by using dissimilar metals, it should not be used in contact, because uh, their half cell potentials are different, and that's why an electrochemical reaction that will be set up between them, which can result in additional polarization, and often in corrosion of one of the metals. And this tends to make the half cell potential less stable, and it contributes to increase the electric noise from the electrode. Okay. Another is in detecting surface potentials on the body, or internal potentials within it, while using pairs of electrode for measuring differential. It is better to use the same material for each electrode, since uh, the half cell potentials are approximately equal. And this implies that net DC potential seen at the input to amplifier connected to these electrodes is relatively small. It may be zero, which minimizes the possible saturation effects in the case of high gain direct coupled amplifiers. Another point is the electrodes fixed on the skin's surface, it have a tendency to come off, and this is due to a loss of effectiveness to the tack of the tape holding them in place. So this will be no problem if electrodes are well designed. That is, a lead wear should be extremely flexible but strong. And the point at which electrode lead wear enters the electrode gets broken due to frequent flexing at this point. And that's why strain relief must be provided at this point. Okay, so a region of insulation that gradually increases from the diameter of the wear to one closer to that of the electrode, it reduces this problem, and it distributes the flexing forces over a greater portion of the wear. Okay. Another problem is the insulation of the lead wear and the electrode. Now the environment around electrodes is very humid or continually soaked in extracellular fluid or even in cleaning solution. Now these insulations are made of polymeric material so that it can absorb water. It is also important that the insulation material used with electrode that should be proper to avoid this such problems. Okay, so that's all for today. If you have any queries, you can ask me. Do you have any queries, any doubts? Hello, do you have any queries, any
No,
doubt?
sir. No, sir.